So this is going to be a little bit, in ways, philosophical. And what I'm going to look at is I'm going to look at how to take assumptions that we see being made in various models in biology and how to reconcile them in sort of ways. How to use CS theory to check, do these assumptions actually correspond to each other, or do they clash? Is there some contradictions? And that's kind of the broad goal. And I'll concentrate on sort of assumptions that are made when we're reasoning about static fitness landscapes, right? And in particular, we'll look at this issue that we've seen come up over and over again, which our originalists were right, and this is the idea that in a rugged fitness landscape, we have local fitness peaks. Somehow magically we get to them quickly, and then our questions are implicitly in the language that we use. How do we go from one local fitness peak to a higher local fitness peak, right? And the question is this at all a reasonable statement? And you can see, for instance, in the previous talk, part one, in a lot of cases, we rested the hypothesis on the fact that you can get to a local fitness peak, right? Is this always the case? Can you always get to a local fitness peak quickly in a static fitness landscape? And again, as was pointed out 2 talks ago, all of these things are more interesting if you look at dynamic fitness landscape, et cetera, but we're looking at the base case of the simplest thing we can do. If you have a more complicated model, you can probably embed these smaller models inside of it and get the hardness results anyways. And that's what we're going to concentrate on, as well as sort of showing that things are hard as opposed to showing that things are easy or doable. And so what do these sort of empirical fitness landscapes look like? Well, the first thing that a lot of biologists care about is this idea of epistasis, right? So you'll have some 2 loci, and they'll have, let's say, alleles A and B, right? You'll have some fitter alleles A, B. And we'll say there's no epistasis, or there's only magnitude epistasis, if either way of getting there, first swapping in A or first swapping in B, are adaptive paths, right? So going from here to here, increase in fitness, going from here to here, increase in fitness, same here, right? And this is sort of your standard case of no epistasis between these 2 loci, and or magnitude epistasis, which it won't really matter for the sort of the 10-mile CS theory perspective, but it does matter in practice, obviously. On the other hand, it might be the case that sometimes one of the paths is blocked, right? We can't swap in B before we swap in A, right? So if we were to swap in A, it's adaptive. But if we were to try to swap in the uppercase B first, we can't. There's a fitness value that we have to walk into, right? And this is called sign epistasis. And of course, there's the worst possible case you can have, which is reciprocal sign epistasis, where both paths are blocked, right? And so if we look at sort of what data we have on empirical fitness landscapes, we can see that there's definitely some cases where everything is smooth. We have a smooth fitness landscape. There is no sign epistasis. There's no reciprocal sign epistasis. You start at the lowest fitness genotype, and then all the alleles are adaptively flipped in, and you can get really quickly to sort of the fittest variant. However, there's also empirical landscapes where you'll have both sign epistasis and reciprocal sign epistasis, right? Like example here, you'll have 2 high fitness genotypes, and everything that's written in red is their shortest adaptive path is to this local fitness peak, and for these guys, everything in blue, their shortest adaptive path is to this local fitness peak, and for the green one, it depends on the particulars of how strong the selection strengths are on those edges, right? So in real fitness landscapes, we kind of see all of these kind of things showing up. And so in particular, we can't always assume smooth fitness landscapes. We definitely can't always assume that there's one peak and we can get to it quickly. In general, we might have several peaks that we have to worry about. And we kind of want to quantify this in a very sort of vague way and see where we can get from it. Another thing to notice from these fitness landscapes is that it's on 4 genes, right? N equals 4. For n equals 4, n squared and 2 to the n are the same thing. these are very, very small examples, which is really hard to distinguish anything on. And sort of the best state-of-the-art experiments done on this, you'll have n equals 6, n equals 8. We really don't have a good idea of detailed fitness landscapes for anything, really.
But I think that's not the E. coli beta-lactamase. I think they were citing that paper. I think this is the Methylobacter.
I might be wrong.
Yeah, I think you might change the reference.
Okay, I might have
yeah.
swapped my references. I'll fix that up. But the idea is just to get you a heuristic idea of what these landscapes look like. And so, since we're looking at these, since we have so little information about landscapes, we have to figure out how to proceed with reasoning about them. And in a lot of modeling, the standard way is to say, okay, well, I'll just pick my landscape randomly. Which is kind of, to me as a computer science, that's a very weird thing to say, because a lot of times picking something randomly gives you a lot of structure in that thing that you've picked, right? And so as a computer scientist, I'm going to have to recreate part of Paul's talk here and talk about worst case analysis. But before I talk about worst case analysis, I'll talk about sort of a biological model which gets an easy way to look at epistasis, right? It's kind of the standard way, an old model to look at epistasis. Probably not the most popular anymore. And probably everyone's familiar with the NK model. Just for those that aren't, what you'll have is you'll have some sort of simple gene interaction network where a given gene interacts with k other genes, right? So I'm drawing 1 for k equals 2, let's say. Right? And what interacts means is its fitness depends on the value of the alleles in the genes that it interacts with. So for instance, the fitness of x0 here depends also on what's put into x1 and x2. And then the fitness of x2 maybe depends on what's, or on x1 depends on what's put into x2 and x3. Right? And your overall fitness is then calculated as a sum over these interactions, right? So. And the genes that it interacts with. Right, so that's an NK model, right? And we'll care in our case about NK models for k equals 0, which means no interaction, everything is independent. That's a smooth fitness landscape. You can just put the advantageous one in each place. Then we'll care about the case n equals 1, which is a case that's tractable, and the case n equals 2, which is the same as all other cases. They're all equally hard. And we're going to show that, for k equals greater than or equal to 2, it's a PLS complete problem. So we need to introduce sort of, we need to justify asymptotic worst case analysis, because that's what we're going to rely on as computer scientists. And this is one place where I think computer scientists have something to offer biology, which it might not be obvious. A lot of physicists come into this field and they'll say, "Oh, random, everything works. I'll bring it over from how I used to do it in statistical mechanics." Computer science have to worry a lot about how worst case analysis relates to things like average case analysis and that mythical typical case analysis, right? Because when you're looking at, let's say, a software product that you've engineered, sometimes maybe the worst case analysis doesn't matter. Maybe the users never input the worst cases. Right? How do you produce? And so computer science have thought a lot about this, and there's definitely all kinds of disagreement on when it works, when it doesn't work. Obviously, I think that worst case analysis can tell us something very useful about fitness landscapes that we couldn't get otherwise. And what it can tell us is, we'll look at the space sort of all fitness landscapes, right? And what we want to identify in there are the ones that are actually present in biology, right? To some extent. And let's say we're married to this idea of having quick paths to local peaks, right? Let's say, I can't live in a world where that doesn't exist, right? Then through worst case analysis, we can say, okay, well, these sort of fitness landscapes over here have exponentially long walks, so you don't want them. They're not part of your typical set, right? Or maybe we're looking at some system where we know things happen extremely quickly, faster than linear, logarithmically fast convergence, right? Like in parts of the immune response, right? Then we'll say, okay, well, that's probably in this set here where we can prove that convergence happens quickly. And all this will come from worst case analysis, will allow us to divide up our space as a sort of first step, right? And then afterwards, once we have some idea of figured out what the space looks like, we can start to say, okay, let's talk about probability distributions over this typical subset that we've identified. So worst case analysis lets us cut down what fitness landscapes we consider to be reasonable fitness landscapes. Okay? So what happens for Kaufman's NK model? Well, now we'll see a series of results which will be the more handles you give me, the more weight I can lift, right? So if you tell me nothing about how mutation works, right? It could be some complicated learning algorithm. At each generation, the mutation operator goes and checks if some numbers are prime, solves a hard linear programming problem, and things like that. If you tell me nothing, but you let me assume that p is not equal to PLS, then in general you will not have any local fitness peak to which you can get in polynomial time. Right? There will be landscapes where that's the case. Now, if you let me be more like some of the biological literature and say, okay, well, I'm an adaptationist, I only care about things that follow adaptive walks, right? Then we don't even need the p not equals to PLS condition, right? If we look in point mutations on this genotype space, then for some conditions and for some landscapes, every adaptive path to any local fitness peak will be of exponential length, right? So it doesn't even matter if you have some sort of...
And actually, from almost all starting points.
Yeah. And it doesn't even matter if you sort of have an omnipotent bacteria rancher who sits there and selects which adaptive mutation gets to come in, right? As long as the mutations have to be adaptive, no matter how much guidance you try to give them, it'll still take you exponentially long to find any local fitness peak, right? So it's a very strong result for artists. So you'll never be at that peak and be able to say things like, I'm at that peak, my mutation will start to drop. But I'll talk more about the approximate peaks that you cared about as well towards the end. But you might say, okay, well, these are pretty complicated landscapes. Can you tell me something stronger for simpler landscapes? Like I think k equals 2 is already a pretty complicated landscape. But even empirically, people have found this distinction that as you go from k equals 1 to k equals 2, everything gets a little bit more complicated. Right? Can you tell me anything simpler? Okay, well, if you give me more handles, if you let me specify more what we mean by how the mutations happen, then I can cut off even more of the landscape space as inaccessible. Right? In particular, we can define this concept of semi-smooth fitness landscapes. Right? So for smooth fitness landscapes, this is the only type of epistasis that happens, magnitude epistasis, no sign flipping. Right? For smooth fitness landscapes, it's easy to see that you can always converge in n steps, right? Because you always put in, and that one doesn't matter which order you go into. If you want to do a more careful analysis instead of steps in terms of time or generations, that'll be a slightly more careful analysis, but it'll be still polynomial, right? If you allow me reciprocal sign epistasis like the NK model does, then I showed that it's hard. So instead, let's say you allow me only sign epistasis on a single gene. It won't be ever on a pair of them, right? So there's always a shortest adaptive path, right? This means it'll be in a unique peak, and it'll always be from anywhere you start. There'll be an adaptive path from there to that fittest peak, which is of Hamming distance between you guys, right? So there'll always be a short path. Well, you won't find it. And this comes from taking results from the previous results were from looking at polynomial local search. These comes from looking at the simplex algorithm. The simplex algorithm, they define these tool to analyze them called AUSOs, acyclic unique sync orientations, which basically correspond to what I just called semi-smooth fitness landscapes. And you can show that, okay, well, if we do that sort of fittest mutant always, right? That's a strategy that happens when you have a very, 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 very large population, right? Then, again, exponentially long walk where you find the fittest mutant, and you'll walk around your hypercube until you finally get to the peak, right? And you might say, okay, well, no one actually used that except for maybe some genetic algorithms, things. In reality, we might use something like strong selection, weak mutation, right, where we pick one of the adaptive guys at random, right? And even there, you can show it's a little bit harder. You can show that with high probability,
the Fisher exponent. The Fisher exponent.
Yes, you'll have the Fisher exponent of higher probability. You'll have your e to the lower bounded by e to the n to the third power to the 1/3. Yep.
Do you know the long path problems by Jeff Horn?
Not off the top of my head.
It's in the GA community, so he showed the fitness landscapes where he'll try to take the exponential time to solve it even though it's single peak. Sounds like it might be the same thing.
Yeah, similar things. And I mean, you can try to look at the ones in between, right? Sometimes people say, okay, well, I'll isolate them in proportion to their selectance strength and things like that. You can look at those variants. Some of these theorems are hard to prove, so it'll be a hard theorem to prove, but there's no real natural reason to expect that things in between, selection rules in between, or in the simplex algorithm community, pivot rules in between, will get you something better. There's no reason to expect that. So in other words, what you happen to give me here, you give me more control over how the mutations happen, and I cut off more of the fitness landscape space on which you can find your fitness peaks, your local fitness peaks efficiently. Okay. So you might say, I don't care about, actually, when I say I'm at a local fitness peak, I don't actually mean I'm at a local fitness peak. That's a straw man. I mean I'm near a local fitness peak in some vague sense of near. Well, there's 3 ways you can be near. Well, at least 3 ways. There's many ways you can define, but there's at least 3 ways that pop into my head that you can be near. Okay? So if by near you mean you're a few adaptive steps away from being at a fitness peak, then no. No, you're not near it in that sense of the word for these hardness results. This is all assuming that PLS is not equal to P. If you mean that your fitness is close to the fitness of a local peak, then let X star there be the fitness peak, the local fitness peak of lowest fitness, and you're not even exponentially close to its fitness.
What's the distribution of fitnesses?
Hmm?
I mean,
The NK model.
NK model. Okay,
Yeah.
so it doesn't matter what distribution I draw.
There's no, remember, we're looking at worst case, so I'm not letting you draw. So what happens in the worst case is I put some specification on this network, and I put specifications on what these additive terms are, and these additive terms are polynomial in whatever's my parameter complexity in the n.
Okay.
Right? But yeah, once you start sampling things, and there's results like, if you sample the network at random, you sample the weights at random, then you get this graph of a lot of local peaks that are really, really easy to get to, right? You can show that models the immune response relatively well, right? But that's really extracting structure from randomness, right? That randomness gives you a lot of structure. Here, it's a worst case, so they're specifically built in such a way that they're hard to find the local peaks. Okay, so those are some sad results, I guess. But there is one way you can have your local fitness peak, and that's to say that if my fitness is f of x, right, and if for all mutants in my neighborhood, we have that my fitness is greater than or equal to 1 minus s of y, right? So in other words, there might be some guys that are fitter than me, but they're only fitter than me by this s amount, right? Letter chosen for suggestive purposes.
Very flat plateau.
Yeah.
That sounds like a plateau. What about another definition of a fitness peak, which is that the fraction of my beneficial mutations has gone to be vanishingly small and become small?
Yeah, so this won't matter because these neighborhood sizes are polynomial. So your sampling is still going to be a polynomial amount of time sampling there, right? So for these exponential results, it doesn't matter that it's small. It'll still be at least 1, right? Unless you're actually at a fitness peak, there'll be at least 1 nearby mutant.
Right.
And since you're sampling, there's n of them, and you can just find 1 in time n, you'll find it, right? So it won't. It's definitely matters in practice, right? Where we care about the difference between do we get there in linear time, do we get there in n to the 17th or something. But in the case of separating exponential from polynomial time, it does not matter.
And it matters for the evolution of mutation.
That certainly does.
What he said, he wants the fraction of steps that are beneficial to decrease, say that to go to a constant independent of n.
Yeah. Or just
Yeah,
you can
that's
do
fine.
over time.
I can assume there's only just 1 of them,
No, he's just wondering, like, is there a way that you can make it so that it takes a very long time to get to that stage, where there's only
I'd have to think about it. I don't know.
Yeah, that's the question.
Okay.
So most of these models, you start out with a certain distribution of beneficials. That distribution will go down, but you're saying it's just never going to go down to 0 or close to 0. So I still think that the fact that it goes way down below some threshold matters.
So sort of. I mean, the other thing that always matters is when they go down is also how big that selection strength gap is.
Yeah.
Right? Because even if you have one guy and he has a very, very high selection coefficient, you have a 1 in n chance of sampling him. He has a high selection coefficient, he'll take over the population, right? And this is where the result starts to matter a little bit, is we can achieve this if in time polynomial in n and 1 over s, but we cannot achieve it in time polynomial in n and the natural logarithm of 1 over s. So why does that second part matter? This second part really matters when we want to look at cases where we actually have data. Like I said in the beginning, we don't have much data on actual structure of the fitness landscape, the combinatorial structure, but we have a lot of data on the time dependence or the fitness time series, right? And the classic example of this is Lansky's long-term evolutionary experiment. So we have data like this, where we have the fitness over some long number of generations, right? And so in terms of this, we can take our equations from the last slide and turn them around. What does it tell us? Well, it tells us that on these hard fitness landscapes, S, which is the gaps between them, between each increase in fitness, can drop off as a polynomial in n and 1 over t, right? But it can't drop off as the exponential in time, right? So these little gaps.
Wait, I feel like there's two quantifiers going opposite directions here that I don't understand. So I could pick a really nice fitness landscape where I could make S drop as fast as I wanted.
Of course, the smooth one will do that. Yeah.
So what's the statement here?
The quantifiers for hard fitness landscapes, so for these hard examples, right? If you allow me all of the NK model for k equals 2, right? On these hard cases, you cannot get exponential convergence towards a local fitness peak, right? And so the nice thing about Lansky's recent result is that this is kind of one of the things that they show, right? They show that you don't get very fast convergence towards an asymptote in their case, but also exponential convergence. Instead, you get this sort of power law conversion, which translates into S dropping off as A over T, where A is the parameter that they get from their fit. So this gets you in a kind of a place where you have a lot more data to play around with and say, okay, well, in here, we're getting this slow increase in fitness. Can that tell us about something about the structure of that fitness landscape potentially? Right? Can it at least help us cut off some of the fitness landscape space?
I don't know. I mean, I don't know how much you want to really trust these functional forms here. It's not a huge range of data.
Yeah. I mean, if we actually look at this carefully, then this line is actually dropping off as S over T squared. So it's not even separating between, they're separating different kinds of polynomials, and they're separating on a very small, not a very large data set. It's meant more as a picture. Right. But it does sort of tell you how you can start to connect to experiments, right? And in particular, as I showed on the first day discussion, one of the ways you could start to test these sort of models, right, is to start running little, Lansky-like experiments, right, where you say, okay, well, I find some, and this is me speaking as I'm not clearly non-biologist, so it probably doesn't work at all, picking some, this gene codes this kind of protein, this gene codes this kind of protein, they're kind of relatively independent. We know that from some other observations. Then put some tracer on them, watch them. If their combination satisfies, some satisfiability problem, then I don't kill them. If it doesn't, I do kill them. And this way I can make this very hard landscape for them to evolve in, and see how fast they evolve in that sort of landscape. On the other hand, give them a simpler one and see if they get the faster scalings.
Why wouldn't I want to do that on a computer?
Because on a computer, you'll get these results for sure. The idea is if you want to look for magic or something.
Oh, okay, yeah.
Right?
If I don't believe in magic.
yeah, on the computer, yeah. So if your half water bottle challenge, right? If you let me encode weighted 2SAT into your difficult problem, then you won't get to local fitness peak, right?
But the fraction could still drop to be so rare that I don't ever generate them, and so I still have the same result.
It'll drop off like this.
I'm sorry, I looked right there.
It'll drop off like the yes. It'll drop off like the yes line, right?
You really like that water bottle.
I really want that water bottle, but you know we can talk about it a
Yeah,
little bit
just
and
cut
see
it up.
if
I
I can
don't
encode
think
it.
it'll work.
Okay, and then where's the case where we can really worry about this? You know, before we kind of worst case analysis, the easiest way for computer science to justify worst case analysis is to say, you know, adversarial, right? An adversary builds my fitness landscape, and that's exactly what we saw earlier today, right? You know, cases like the immune system, where the host is picking the fitness landscape for the pathogen to adapt on, and it's picking the worst one possible.
But
Right.
he's picking it by volume.
Hmm?
So, but he's picking, but he's adapting into it. I mean, also.
Yeah. There's restriction on how it's picking, but that comes up to still the computer science problem. Can you generate hard instances of problems that you then can't solve, right?
Right, but,
Um,
but here, sort of, you have two adversaries with their hands tied behind their backs.
To some extent, yeah. But in these cases, I think you can make a much better justification for why
No, agreed.
the landscape would be the worst case possible. And then this might get you some maybe intuition about cases like, once again, I don't know anything about biology, but in cases like, you know, let's say HIV, where your genotype is constantly changing, right? Can you look at those things as artifacts of the fact that the immune system builds a fitness landscape for that virus, which it can never find a local optimum, so it keeps chasing something constantly.
That one, it's a shifting landscape. So.
Okay.
But yeah, along those lines.
Right, and for examples like that, and see if that's a useful thing to think about. And so I guess the whole point of the talk is to sort of figure out how, if we take our assumptions, we make them very precise, do they always add up? Not really. In this case, you know, if you like the NK model for some reason, but you also like the idea of finding your local fitness peaks quickly, you have to cut off parts of the possible fitness landscapes in order for this to work, right? Or if you like your strong selection, weak mutation model of adaptation, right? Then you really need to cut off huge parts of your fitness landscape for them to find fitness peaks quickly. So yeah.
Would you say cut off huge parts of the fitness landscapes?
Yes. So like that result on the previous page were semi-smooth fitness landscapes, and you still get hard walks, right?
Mm-hmm.
So that already tells you that there's this.
It's some. That's still worst case.
Everything is worst case.
And are some of these where the worst case has some sizable fraction that's only like decreasing polynomially with the size, or
What do you mean sizable fraction of what?
like where like a polynomially small fraction of the total number of landscapes?
So this is where we're starting to get into average case analysis, right? So when you start talking about fractions, right, what you said is I assume the uniform distribution over fitness landscapes, and if I sample from the uniform distribution over fitness landscapes, do I have a non-zero probability chance of sampling this
It's
one?
asking a natural question. How, and the answer is we don't know. We have a huge space. We say, look at that, look at that, look at that, look at that. For all we know, it's half of it. I mean, we doubt it, but yeah.
I mean, we can definitely say, for instance, there's more than polynomial. Many hard instances, because you can take one hard instance, kind of padded, for a slightly bigger hard instance, and then you have a lot of hard instances of a slightly bigger size.
But exponentials,
You're saying the semi-smooth.
And over 100, who cares? Yes,
You're saying that if you restrict it to the class of landscapes which are semi-smooth, which is a small fraction of the space, there are still ones that you can't guarantee to solve.
Yes. And you can blow those up also to be not like a tiny fraction by padding sort of arguments. But you can't say
Not a tiny fraction.
So not a tiny fraction, as in like more than, not a sparse set, so more than polynomially many of them, right?
More than
Polynomially many of them. But that doesn't get
the
you.
space is exponentially big.
The space is exponentially big. You have more than polynomially many of them. Doesn't actually mean that you have a non-zero probability of sampling them, right? Once again, because exponentials are mean to me. But the whole point of this that I was trying to make in the beginning is that these arguments, when you ask this, what fraction of them, you're implicitly asking me to put a probability distribution on the space, right? And you're asking me to put the uniform one. And the idea is that if this static fitness landscape, if it's coming from some ecology or something that the organism lives in, there's no reason to assume that those ecologies are somehow sampled randomly.
I'm asking if you need, if I want to find these worst cases, if I need to assume some really extreme probability distribution.
It's hard to say. You're asking me for the mythical typical case. And then the whole point of this is to kind of help identify what that typical case is by cutting off parts of the fitness landscape space. Yeah. All right, thank you.